Hello, good afternoon. Love to see such a large and wonderful crowd for cystic fibrosis-related diabetes talk. So welcome, yay. Scott and I are gonna be the chairs for this session today. We both have nothing to disclose. And we just want to talk a little bit, give a little background on cystic fibrosis-related diabetes. Some of you may not know that cystic fibrosis-related diabetes is a relatively uncommon diagnosis in the first decade of life. But we do know that glucose abnormalities begin within early childhood. By the time that we reach to the second decade of life, about 20% of adolescents will have a diagnosis of cystic fibrosis-related diabetes. And about 50% of adults who are over 30 will have a diagnosis of cystic fibrosis-related diabetes. There's increased morbidity and mortality that is associated with this diagnosis. Unfortunately, mechanisms that are involved in the development of cystic fibrosis-related diabetes are not completely understood. For many years, we attributed CFRD to the loss of islet cell pancreatic architecture. Secondary loss, secondary to exocrine pancreatic damage. But post-mortem studies tell us that is not the complete story of cystic fibrosis-related diabetes. We do see that individuals who have the diagnosis have insulin secretion defects for the islet cells that are remaining. And along with that comes an impaired first phase response to glucose. Other mechanisms that may be involved, and further research is definitely needed within these areas, would be secondary factors such as having a family member who has a diagnosis of CFRD, abnormalities, or having a diagnosis of type 2 diabetes, abnormalities within CFTR, whether they're an impact from actually being in the islet cell versus being in a bad neighborhood, from the exocrine pancreatic damage, inflammation, and then the impact of nutrition. Today, we have presenters who are basic scientists and clinical researchers that are going to address a broad variety of topics within this area. We're going to start with Xingshen Sun from University of Iowa, and he will look at the effect of ivacaftor on pancreas in the CFTR G551D ferrets. Rebecca Hull from University of Washington will discuss CFRD, a disease of the pancreas microenvironment. Eddie James from Benaroya Research Institute will be looking at investigating beta cell reactive T cell and immune phenotypes in CFRD. And Katie Larson Ode from University of Iowa will discuss the Envision CF multi-center study of glucose tolerance in CF.
And it should also be on your program, but we have also scheduled in a panel discussion at the end, so we hope if you have the opportunity, you'll stay to the end and ask all of our experts whatever is on your mind about CFRD or pretty much anything else.
So our first speaker is Xingshen Sun from University of Iowa.
Hi, good afternoon. I'm Xingshen from University of Iowa. Today I'd like to thank the organizer, the CFRD session, for the opportunity to share our recent studies about the effect of ivacaftor on pancreas function in G551D ferret. Nothing to close. And this slide shows the outline where I will talk about today. I will share some of our recent findings in the long-term effect of ivacaftor on the pancreas. Then I will talk about affect disable CFTR person at a different development age on the glycemic control. And the last, I will shared my studies on the recovery of the exocrine pancreas. So when you read title, maybe you think, why the ferret? And let me give you some little bit description and what I think for why we use CFRD models for the people who's not very familiar with CFRD. And we use ferret as model mainly because the CF models capture many CF pathologies observed in humans in multiple organs, especially the pancreas and the pulmonary. And you can see from this slide in the left, it has the human pancreas and the CF ferret pancreas share the similar pathologies and lost acinar cell and replaced by the fat tissue and the fibrosis, and important endocrine elite structure architecture is totally distressed and abnormal look like. And also they share the same functionally the pancreas insufficiency and glucose glucose control in the early age, and later on they will develop diabetes. The right figure, the top one is the human people with cystic fibrosis have the higher level the glucose levels response intolerance, and the bottom line you can see the CF ferret share the same the glucose intolerance. So more important one thing is we have not just the CFTR models, KO models, we have another two CFTR modulator response models, G51D and 508 delta. And combined with the CF modulator, I think these models combined with the CF modulator provides us a unique opportunity to study how the CFTR expression affect glucose control, because we can turn on and turn off the CFTR expression by start avicaptor treatment or stop the avicaptor treatment and or restart again. So we can play around. So this is the short study we published several years ago. This we think because the CF ferret have similar to humans, the CF pancreas also the pathology start in uterine. So we reason that if we want to maintain a normal pancreas functions, the re-expression CFTR should start as early as possible, and postnatally should maintain the CFTR expression as longer as possible. This one is the short study. We studied 3 or 5 months to give the G51 ferret avicaptor in uterine, and up to 5 months we monitored the glucose, postprandial glucose, and gave glucose challenge test. And this result is we can see the administration avicaptor to the CF ferret in uterine and postnatally not only maintain the prevent CF pancreas pathology, maintain the pancreas sufficiency, also improve the glucose regulation. You can see from this slide that the left one is the postprandial glucose fasting and after feeding, no significant difference between the wild type and the treatment group. And most obvious one is the when they perform the OGTT, the time course response is the same, no difference can see. So that means they can provide improved glucose tolerance. So one question we want to ask or want to know is what's the long-term effect of avicaptor on glucose? In other words, if whether we need if we want to keep the pancreas function, whether they need give the avicaptor or express CFTR the whole life. So this thanks for CI Foundation invite money for us to support our longitude study. So this year we enrolled 5 G51D ferrets. 8 they have been treated avicaptor from the start from uterine for more than 2 years. So we try to see whether they have a normal glucose tolerance or normal glucose. So first we tracked the pancreas damage and measured the E1 level. We found that 80% of them have the fecal elasticity 1 about 200, the threshold for the insufficiency and the insufficiency. It's very hard to pronounce the word. And only one animal is pancreas, exocrine pancreas insufficient. So these animals, when give the glucose ordinal test, we found the ferret with pancreas sufficient one have normal glucose tolerance. But the animals with the pancreas insufficient one, they have peak levels at 6 minutes about nearly 300. That means the glucose control is abnormal. So even we need we will do more experiment, enroll more animals to confirm these observations. But they clearly see that the continuous helicopter treatment will need to maintain the glucose tolerance. At the same time, this glucose tolerance maintained only in animal with pancreas deficiency. So another question is because we find have 20% pancreas deficiency, we don't know when and where it's happened, because this one can happen at any time during the helicopter treatment, neither the neonatal stage or the young adult stage. So this is the second question we want to figure out. So we use G5 to design the second experiment, discontinue the helicopter treatment at 1 month right before the weaning, or the 4 months at the young adult stage. We enrolled the T5ND homozygous ferret maintain the helicopter treatment for 1 month or 4 months, and then withdraw them. After withdrawal, remove the helicopter. We weekly monitor the postprandial glucose, and at the 2 months or 5 months, perform the oral glucose tolerance test. So from the left figure, you can see we first check the pancreas damage and measure the E1. You can see after remove the pancreas helicopter, the pancreas damage happened very quickly. In 2 weeks after removal, the E1 level almost reached a drop to 0. So that means the CFTR very important to maintain the pancreas architectures. Even these two groups, the pancreas damage, the rate is very similar, but the glucose alternation level is different. From the right figure, you can see in the early group, after remove the helicopter through 3 to 6 weeks, they develop acute hyperglycemia at the peak at about more than 350, and followed by a recovery after 6 weeks, remove the helicopter. In adult one, there's no these things happens. There's no developed acute pancreas hyperglycemia levels. So then we again give the oral glucose test at the recover stage. This green part, this one green part one, this not work. And you guys see at the recover stage, it's not surprising that in the early group they have normal glucose tolerance, but later group, after 2 months, removed the glucose level is very high. It's abnormal glucose tolerance. The overall glucose levels also, the later group is much higher than the early group. We see the OGTT category, the right figure. You can see the most early group one have the normal glucose tolerance, but the later group, they rapidly developed impaired glucose tolerance test and the CFRD. But after 5 months, both group, neither the early group and later group, both have the glucose intolerance. And the OGTT category in the early group shifted from left to right, so that means some normal glucose tolerance tests became abnormal. So this study suggests that the CF pancreas during the weaning stage may have a great ability to recover elite function in face the exocrine pancreas destruction. This kind of ability declined with the aging. This figure is pretty similar to the last slide, the right one. So, but it must clear to see how the early group and the later group change the OGTT category. From this figure, we can see the later onset CF damage progress to CFRD much faster than the early onset CF damage. Then we do the pathohistology analysis to see what's the eyelid change during this short time the damage. You can see the early group and the later group CFD, figure CFD is significant different in the morphology. The later group, the eyelid became the diffuse and became scatter and small eyelid. And early group 1 have the larger cluster and the eyelid, the morphology. We qualify the errors when compare, because this we not compare with wild type, but compare the two early and the later. They both the exocrine damage have been lost as the cell, so it's much easier to compare. You can see the early group, they have much more the beta cells, the error percent. This quantification suggests that eyelid Spanish occurred in G5 ferret following the crisis. This process appeared to be age-dependent in G5 ferret. So, this study is go back to our KO studies. It's pretty similar. The result consistent with previous findings published in 2016. We used the CFKO ferret. We found CFKO ferret go through the same glucose natural history change, abnormal change. They had a winning stage, have a crisis stage, the developed acute hyperglycemia postprandial, and then followed at recovery stage. When we do the dual nucleotide labeling, we find in the late stage, the eyelid cells, they have regeneration. So that's maybe why the in early group have the large eyelid and have the higher percent incident cell error. That's also maybe contribute to recovery or delay the CFRD in the early group. The third question we want us to know, we assume that in the this stage, whether the exocrine pancreas as the endocrine cells also have retained regeneration ability. So let me just give unexpected findings. When we do the continuous and discontinuous experiment in G5-1D, we find even the continuous group ferrets give the whole life the avicaptor expressing CFTR. We find we measure the E1 level to monitor the pancreas damage. We found at the weaning and the young dog stage. In the weaning stage, it's pretty critical. We found some of the ferret is even give the continuous avicaptor. They still have some ferret is developed in pancreas insufficient. But when we remeasure these animals, we found after 1 month or 6 weeks, they go back again to become from the pancreas insufficient become pancreas sufficient. This one finding. And another case report, there are many case reports about avicaptor can recover the exocrine pancreas. If you remember me last year, I remember a speaker report this one, rival trials. They find gave the young children at 12 to 24 months old, similar to the weaning stage in the ferret. If they have reserved acinar cell and a little bit baseline E1 level, continue give the avicaptor can recover the exocrine pancreas. So we hypothesis that the exocrine pancreas at this early stage, similar to the endocrine cells, they still retain the regeneration ability. So we designed this experiment. Baseline basically the same in the early group, but just after 1 month, turn off the CFTR expression, then we go back on the avicaptor to turn on the pancreas CFTR expression. we find that in the left figure, before the turn off the CFTR expression, the pancreas is sufficient. And after remove the helicopter, turn off the CFTR, in 1 month, the pancreas dropped to nearly to zero, but they still have a little baseline. About 3 months later, so they recover again. So this clearly demonstrates that in this stage, pancreas will still have the ability to regeneration. When they perform the OGTT in these animals, pancreas sufficient 1 and the pancreas insufficient 1, and the glucose level is much better in pancreas sufficient 1, but the insufficient 1 is very high peak level in the 6 minutes. The overall glucose also significantly higher in the is lower in the full recovery 1. The E1 have high level 1 recovered. And when we correlate this response, we can see neither you correlate with the glucose overall glucose levels or the peak levels at the 6 minutes. The E1 level higher, they have inverse the correlation. In the high level E1, they have lower glucose AOC and lower peak levels. So that means suggests that the level of biomarker pancreatic sufficient is inversely correlated with the risk of later CFTRD. The fourth question we want to ask is, what's the mechanism of regeneration for exocrine pancreas? So this one is the immunostaining for the pancreas, ferret pancreas after turn off the VSM helicopter, turn off the CFTR version at 1 month. You can see most of the acinar cells, the red one is lost. The MIP7, we mark the dark cells. They have the overgrowth, the pancreatic cell, and also have a lot of cells have the core-standing, the acinar cell and the dark cell markers. So we hypothesize that during the pancreas damage and the CFTR and lost expression can cause the inflammation and cause some acinar cell transdifferentiation into the dark cells. And when the inflammation resolved by turn on of CFTR, maybe these some cells can turn back to normal acinar cells to function, but we don't know what the cell is come from. Maybe the core-standing cell or existing the stem cell or progenitor cells, so medically. But I think this question to be answered have to go to the linear tracing studies. This study we have not done yet, so it's just our future studies. So in summary, the continuous helicopter improve glucose control and delay the PRO1 CFRD, but only in animals that are pancreas sufficient 1. Discontinue the helicopter, turn off the CFTR treatment, lead to the exon pancreas insufficient and disturb the glucose homeostasis. And the OGTT category are determined by the timing of CFTR expression when it's turned off. And the pancreas at the early stage, especially the waning stage, may have a unique capacity to regenerate both endocrine and exocrine cells. Identifying the source cell for the regeneration, the pancreas cells, neither the exocrine, especially exocrine cells, acinar cell, require and the future study using the linear tracing technologies. So in the last, I will thank my the lab and this study also supported by funding from CI Foundation and Nature Ferret Research Center in hosted in the University of Iowa and directed by the John Hart. Okay, thank you for attention. Yeah.
Thank you, Xingshen.
Yep.
That was a beautiful presentation, thank you. I have 2 questions about the study that you showed at the end, where you just, Rebecca Hull from Seattle. So the study that you showed at the end, where you just withdraw the ivacaftor from 1 to 2 months,
Yeah.
and you show that beautiful recovery of the exocrine function, and then you showed the glucose. Oh, thank you. If you go
Yeah.
back, so
Yeah.
here, okay. So my question, and I don't work with ferrets, so this might not be possible in this animal model. Have you done OGTTs? Have you done more than one OGTT in animals over time? So if you look at the OGTT, for example, at 2 or 3 months, when the exocrine insufficiency is present, and then look to see if the animals recover, can you look at the time course of glucose tolerance over that time?
Yeah, we plan to do that, but this one's only one time. You can see here is not fully recovered IgD. Look like a little bit impaired. We hopefully in the second or third, the endocrine function can fully recover. Yeah.
And what time point did you do that OGTT? I'm sorry if I missed it. When did you do the OGTT?
The 6 months.
Okay.
Yeah, 6 months. Yeah.
Okay. Sorry. And then the other question is same study, but I'm wondering if you have you looked at the exocrine pancreas morphology or pathology just at the end of the time when you take them off ivacaftor? So really my question is, do you know that the exocrine tissue is completely destroyed?
I don't know yet.
Okay.
Don't know yet.
Okay.
Yeah, hopefully we will enroll more animals to do more the histopathology. Dentist. Yeah.
Yeah, it's fantastic work. Thank you.
Thank you.
So a question for you. I may not have seen it, but I was just curious if you have a model in which you had not treated any of them in utero and then started treatment at a later date, sort of mimicking the way a person might see ivacaftor.
Yeah, that's good questions. Yeah, this one the technique problem is if without a uterine treatment, the afterborn they mostly cannot survive, because that's why we give interior and 1 month we off them. So we hopefully they can survive or not. Yeah. Okay. Thank you.
And I just want to remind everyone, if you don't want to come to the mic, you can log into the app and submit your questions. I just looked, I didn't see any questions. Oh, there. Thank you, Tony.
I have a quick question. Tony Moran, Minneapolis. I might have missed this, but when in utero did you start the treatment? I mean, what if you had started before they even got pregnant? Or is there some critical period that maybe you missed?
Yeah, we have the 2 treatments. One is at in a ferret generation is 6 weeks. So we start treatment at 3 weeks, at 21 days. And sometimes we treatment at 28 days, the 4th week. But when we do the IR-seq in the neonatal of the fetus pancreas, we find that at 21 days pancreas, the CFTR have the expression there, the duct cell have expression. So we mostly start treatment the 3rd week. Yeah. Okay.
Thank you.
Thank you.
Thank you.
So much.
Next, we'll have Rebecca from University of Washington do her talk. Here you go.
Could you start the timer for these? Yeah. Awesome. Okay. Good afternoon, everybody. It's a pleasure to be here. Thank you to Tanisha and Scott for the invitation. I'm really happy to be here to present our work today. I have no disclosures. And I don't really need to go back through this slide because Tanisha did such a fantastic job of describing the state of CFRD. So I'm going to skip over that so we can get into the meat of the talk. And so Xingshen really also did a fantastic job of comparing the pathology in his lovely ferret model to what we see in humans. And so I really just want to briefly point out the fact that when we think about what's going on in CF, we need to think about, with the CF-related diabetes, we really think about the islet, and I'm going to talk a lot about the islet today. But we have to keep in mind the fact that this is occurring, by and large, in the presence of this destruction of the exocrine pancreas. And so I just want to kind of set the scene, and I'm going to come back to this toward the end of the talk. As Tanisha also mentioned, but I'm going to kind of go through this in a little bit more detail, we know that the main defect that we see in people with CF-related diabetes, or even actually within CF at all, in terms of glucose metabolism abnormalities, is an impairment in insulin release. I don't want to say that there isn't any insulin resistance. There certainly can be, but I think it's clear that this is a primary defect. So this is mixed meal tolerance test data from the PEN group. You can see on the left-hand side, see if I can get my pointer working here. On the left-hand side, you can see glucose levels, the gray shaded area and normal ranges. And in people with CF, regardless of their pancreatic insufficiency status, there's this early abnormality in glucose tolerance, right? So both groups have impaired glucose tolerance, with those with pancreatic insufficient CF having greater excursions. When we look at the insulin secretion that is stimulated by this meal tolerance test, you can see relative to our normal controls, again in this gray shaded area, there isn't much of a defect in the people with pancreatic sufficient CF, although their insulin response is certainly delayed. And you can see, of course, that their glucose is not normal. So this doesn't seem to be enough insulin to keep the glucose down. It's important to recognize. But really, when we look at pancreatic insufficient CF, we can see this robust or perturbed decrease in insulin secretion in the early phase and really a profound delayed response. So this is really abnormal insulin secretion. We're fortunate to have had this wonderful study in the literature for a few years now from the Vanderbilt group and others where they were able to take isolated islets from CF donors. This work was done by Rita Bettini in Pittsburgh, and she's really an expert in this group. And so here we're looking at insulin secretion over time. On the left-hand side, you can see individual subjects. So these were 5 donors. You can see their mutation status are listed on the left. And each trace is shown individually underneath. So the insulin secretion is assessed under basal conditions. The response to glucose is shown here. And then a response to glucose plus the cyclic AMP activator IBMX. So this is more of a maximal response, and then back again to basal. And this is collated on the right. So normal subjects have a normal biphasic insulin response to glucose, and then this large response here. And you can see there's definitely an impairment in insulin secretion when we take the islets out. So this isn't only seen in vivo, there's also a decrease in insulin secretion in vitro. And this occurs in the presence of a modest reduction in insulin content within those islets. Although I would say it's interesting to me that you can see that they can respond quite normally to the combination of glucose and IBMX. And so the defect is a little bit more subtle than you might expect, actually. So why is this decreased? Is this decreased when we look in vivo? Is the insulin insufficiency due to the fact that the beta cells are gone? That's really the first most obvious question. And so these data on the left are from our group. And here we were really fortunate to work with both adult and pediatric autopsy services to get a really big cohort of postmortem tissue. And because we had the ability to look both in people with CF with no diabetes in green and CF-related diabetes in purple, you can see that the ages of these two cohorts were quite different. And so we recruited a group of age-matched younger controls and older controls. So the first thing you can see here is how heterogeneous beta cell area measured as insulin positive area per tissue is. That's normal. That's what we expect. This isn't because we can't measure this. This is actually what we expect to see. And you can see that there really aren't any consistent differences among the groups, whether you look between CF and its matched control or between CF and CF-related diabetes. There are data in the literature that don't match ours, and so I want to be even-handed and show that. Marika Bogdani had a really nice cohort where she was able to see very young cases and see that there's a decrease in beta cell area there, and she didn't see a decrease in beta cell area in her adult population, similar to our findings. The heart study, which I showed you data from on the previous slide, showed that actually in their hands there was a decrease in beta cell area. So the literature is a little bit mixed. And I would say in general, there might be some decrease in beta cell area, but it's not completely gone, right? So there are still residual beta cells. And that's a positive because it means maybe we can target these remaining beta cells and treat them and help to prevent and alleviate CF-related diabetes. So then what is the mechanism? What might be going on here? If the beta cells are still there, why are they not secreting insulin? And so the first place to think about this is actually looking within the beta cell itself. And so I'm going to present a little bit of data here. I know that this is a contentious area and that people have clearly shown that there may be a subset of beta cells that make CFTR messenger protein. But there's also quite a large literature that suggests that CFTR mRNA, shown here in brown, and CFTR protein, shown here in green, are by and large absent from the islet. Certainly a few cells might make them, but most of the cells do not. If we look at this panel underneath, the electrophysiology, this is done by Pat McDonald's group at the University of Alberta. Here they have a positive control cell line where they've transfected in CFTR, and they can show this chloride current with forskolin treatment inhibited with the CFTR inhibitor, but they see no evidence for such a current in human beta cells, suggesting again the majority of beta cells don't seem to have much CFTR activity. And actually that's consistent with the data here on the right, where if you take isolated human islets and you treat them with one of several CFTR modulators, there's no effect to perturb or improve insulin release in response either to glucose or glucose plus forskolin. Okay, so that suggests that, you know, there may be some effect in the beta cell, we can't rule that out, but it doesn't seem to be a major effect. And lastly, in this top right panel, this is bulk RNA-seq data from islets from the same CF donors, again from the Hart paper. And here what they're showing is a sort of a selected group of key beta cell identity genes and alpha cell identity genes. I'll come back to these a little bit later in the talk. Their criteria for differential regulation was plus or minus 1 log 2-fold change. And so they see these classical markers, which we know are downregulated in many other forms of diabetes, are really not changed very much in CF at all, suggesting that the beta cells might still be there. They may, but don't seem to be largely affected by direct effects of CFTR expression, and their identity markers still seem to be intact, okay? So what I want to talk to you about for the rest of the talk is that I think it's really important to consider the beta cell in its environment, both within the islet and largely without in the exocrine pancreas as well. And the first part of that talk, the first part of this story that I'm going to talk to you about is the paracrine regulation among the different islet endocrine cell types. So we've talked so far about the beta cell, shown here. I'm going to talk to you next about the alpha cell. The alpha cell makes glucagon, but it also makes a bunch of other things. And beautiful old literature and newer works show that there are positive effects of alpha cell products on beta cells. I can direct you to that literature afterwards if you're interested. It's really, really nicely demonstrated. The delta cell in the islet makes somatostatin, which is classically known to be a paracrine inhibitor of both alpha cell and beta cell function. So what happens to these cell types in CF, and can this provide an additional mechanism that helps us understand what happens to insulin secretion? So first, let's look at the alpha cell. So these are data from our study, but reveal, I think, pretty well what the rest of the field has seen too. So exactly the same subjects I showed you for the beta cell area. Here, I'm missing an asterisk, sorry. Here we see an increase. Something wrong with my statistics here. This is an increase. This is an increase, and this is an increase. So we see an increase in glucagon area. It's different from what we saw on the beta cells, right? And this doesn't seem to matter whether or not you have a diagnosis of diabetes. It's increased in both groups, okay? But then data here on the right from Tony Moran and Paul Robertson. I think this is an old study for some of the younger people in the audience, but still one of the best ones in my opinion. Here they looked at glucagon release in response to arginine. And this is in control, so people without CF, people with normal exocrine function, exocrine insufficiency, and exocrine insufficiency in insulin treatment. And so you can see it's pretty similar to what we see with insulin release, right? So you can see that there's a decrease in all of the CF groups, but a more profound decrease in those of pancreatic insufficiency, okay? So here we're seeing a disconnect, more alpha cells, but less glucagon secretion. Turns out there's other things that might be going on with the alpha cell as well that make it really quite an interesting candidate molecule to study more and try and sort of see if there are treatments that we can come up with that target the alpha cell. On the left, these correlation analyses come from our study, from our histopathology study that we did. And here what we're looking at in these same 4 groups of subjects are the relationship between how much insulin is in the pancreas by immunohistochemistry and how much glucagon is there. So you would expect, as somebody has a greater beta cell mass, they should have a greater alpha cell mass, right? The morphology of the islet is really carefully controlled, and you can really nicely see in our younger controls in blue and our older controls in red that there's this really tight relationship. Super interestingly, we still this relationship is still maintained in people with CF without diabetes, right? So it's actually a little bit surprising to me. Oops. sorry. So you can see that the axis here is different because, as I said before, there's more glucagon area, right? But there's still a relationship. If you have more glucagon area, you tend to have more insulin area as well. Whereas in the purple group with diabetes, this relationship is totally gone, right? There's something completely abnormal about, you can have a lot of glucagon, but not very much insulin. And this comes back to exactly what we see in the ferret model as well, where there's disorganization of the islet. And you can imagine that if the alpha cell is talking to the beta cell, if the islets are totally disorganized, that's not going to happen properly. Okay. Similarly, when we go back to this RNA-seq, you can see this is the only one of these molecules which is actually significantly differentially regulated. It's downregulated, and it encodes a gene called ARX, which is one of the most important alpha cell identity markers. So it suggests that maybe the alpha cell might be more abnormal than the beta cell, and its ability to control the beta cell may be part of what's going on in terms of CF-related diabetes, certainly for more study. The delta cell, I'm only going to show you this one little bit of data so that I can get to the rest of the talk as well. Here we see, and so our data is actually not statistically significant, but the other studies that looked at delta cells are really old, and I couldn't find the papers online this morning, and so sorry for not showing you other people's data. So the literature suggests that delta cells are also increased within the islet. Okay, and if that's true, one could envisage the local release of somatostatin could be increasing the inhibition to both insulin and glucagon release. This has not been studied at all. Somatostatin release in the islet is hard to do. There are groups that can do it, and I think that this is a question that remains unanswered. So I'm going to talk to you for a little while now about some of the cells that are not endocrine cells in the pancreas. And our lab has been interested in the capillary network in the islet for quite a number of years. And so we know the islet is an endocrine organ, right? So it takes those hormones that we've been talking about and puts them straight into the bloodstream. So the vasculature is important both for delivery of endocrine hormones and also for delivery of nutrients, oxygen, and other things to the islet cells. We also know that it's important in terms of survival signals and other paracrine interactions that occur between the endocrine cells and the endothelial cells. I'm not going to go through all of the details here, but suffice to say that the health of both of these cell types is really important in maintaining the health of the other cell types. And we have lots of evidence for this in non-CF disease. So this is a simple experiment. Amy Liu, who was in the lab many years ago now, did this experiment simply where you take islets. This is mouse islets, but we've done the same thing with human islets. The blue figures on the left show what happens if you take these islets just under normal tissue culture conditions and you treat them with low and high glucose. You get this glucose-stimulated insulin secretion, as you expect. And if you do the same experiment, but you then expose the islets to secreted factors from islet endothelial cells, you see this beautiful augmentation in insulin release, okay? Now, if we do the same experiment, in this case, both islet groups were exposed to secreted factors from islet endothelial cells, but the endothelial cells were exposed to either control or diabetic conditions. And here you can see that diabetic islet endothelial cells are no longer able to support insulin secretion, okay? So why am I telling you all this? Well, we think that there may be something that's going wrong with the endothelial cells in CF. Okay, so to contextualize what we're seeing in CF, I just want to show you this data from type 1 and type 2 diabetes. So Eddie James, who's going to talk next, has worked a lot in type 1 diabetes and the autoimmune destruction of the endocrine or islet cells, islet beta cells, right? And so there you're losing your beta cells. In type 2 diabetes on the right, the beta cells are largely still there. And yet, even though the islets look very different in these two forms of diabetes, the vessels look pretty similar. We see an increase in vascular density in the islets. That's the plots in the middle. On the top, we're looking at type 1 diabetes. On the bottom, we're looking at type 2. And we don't know exactly what this means. Is it that the vessels are fragmented? Are they trying to kind of grow and adapt to the fact that the insulin secretion is impaired? We're not sure about that. But it's clear that the direction is pretty similar, despite the fact that the two forms of diabetes are different. So then when we look at CF, we see something completely different from type 1 or type 2 diabetes. And this is one of my favorite slides that reminds me that CF-related diabetes is its own disease. It's not type 2 diabetes or type 1 diabetes in people with CF. It's something very different. And so here, Josh Castillo, when he was in the lab, did a pretty painstaking analysis of looking at the vasculature within the islets. So in the immunohistochemistry panels on the left, we're staining vasculature with CD31. On the top, I showed you where the islet is because it's a little bit hard to see. On the bottom, CF, you can see exactly where the islets are because the whole exocrine pancreas is gone, right? And so when you count this, you look at the amount of CD31 positive area in the islet or the exocrine pancreas, and it's decreased regardless of whether or not you have CF-related diabetes or just CF by itself. We see some variability, right? These guys up here. This case is an infant that is still pancreatic sufficient. Super interesting. This case is a case that has like a boatload of islets, and the islet vasculature looks really normal, and I don't know why. It's like messing up my statistics and my beautiful message here, but it's like, it shows you that CF is really a heterogeneous disease, and there's still so much we don't know about it. The same thing you might expect, because the exocrine pancreas is so messed up. We're also losing vasculature in the exocrine pancreas. That doesn't happen, I don't think, in type 1 or type 2 diabetes. And again, we have these couple of outliers that we're going to go back and study in more detail. So if the vessels are not there, that might even by itself explain why the insulin and glucagon release is not getting out into the peripheral circulation, right? I wish I could say, oh, we should treat people with CF with this drug to help with that. And I think it's not going to be as easy of a question, but I think it's important that we think about this whole kind of environment when we're thinking about the disease. Additionally, well, we talked a lot about the fact that the beta cell and even some of the alpha cell identity genes didn't change very much, right, in CF donor islets. These genes, which are all inflammatory genes, are markedly upregulated. And amongst these genes are some of our favorite genes, E-selectin, IL-6, and others that are really sort of indicative of endothelial cell inflammation. So what I think we're seeing here is that we're losing vessels from the islet, but the ones that remained are probably highly inflamed, right? So there's two ways that could be bad for insulin secretion. I'm going to skip through the next couple of slides just to tell you that there are other things which are kind of unusual about the islet in CF. One of them is that the islet macrophages are gone, right? That's not normal. And I'm going to come back to that when I build my model in just a minute. And we have T lymphocytes, which are not normally very abundant in the islets, are increased. And I'm going to skip over this slide because Eddie James, who's going to talk next, is going to talk all about T cells, and he's way more qualified to do that than I am. So I'm going to just sort of tantalizingly tell you that these T cells seem to be present in CF islets, right? What are they doing there? That maybe is something where it overlaps with the pathology of type 1 diabetes. And I mentioned I was going to come back to the exocrine pancreas because I think we mustn't forget about this. We know that the acinar cells are replaced by fat. These fat cells are probably making cytokines that by themselves are bad for the islet. And we know that the ductal cells, even though we kind of think of them as being out there away from the islet, sorry, this doesn't transmit very well. We know, and we're actually doing an analysis to see whether or not we still see any kind of ductal cells that are around the islets. This is a non-CF case. But in some of our CF cases, we still can find some ductal cells, and they look like they may also be near the islet. We know from really nice work from AP Naran's group that if you co-culture pancreatic ductal epithelial cells with islets and then you inhibit CFTR, you can lose insulin release. And we know from really nice data from the Engelhardt group that if you look at the secreted proteome from CF ferrets, they're making a whole bunch of pro-inflammatory molecules, and they're secreting less kind of supportive factors. So there's sort of a mechanistic sort of hint that these cells are making inflammatory molecules that could also impair insulin secretion. So let me try and put this all together for you then. So here's our beta cell. We know it's fundamentally important in glucose control in all forms of diabetes. I've shown you that there's paracrine signaling under normal conditions between the different endocrine cell types. I didn't talk about gamma or epsilon cells, but they play an important role too. We know the microvasculature and macrophages that I didn't really get a chance to talk about. Both provide important survival and functional cues for the beta cell. There are autonomic nerve fibers that I also didn't get a chance to talk about today, but they impact multiple different endocrine cell types. And there's evidence that the ductal and acinar cells and stellate cells can themselves be beneficial for the beta cell under normal conditions. So in CF, the beta cells are decreased. The alpha cells, delta cells, and I didn't show you, but the PP gamma cells are all increased We don't know about the epsilon cells yet. Microvasculature and macrophages are decreased, okay? The autonomic nerve fibers are likely decreased. We don't know about this in human pancreas yet, but the animal model suggests that they will be. And what we've seen with the vasculature suggests that's also going to be the case. We see amyloid, that's been classically described, which can be bad for the beta cell. But that's only present in people with diabetes, not just CF by itself. And T cells are increased. And then we have this profound exocrine pancreas pathology. So together, I'd argue that we get a decrease in normal stimulatory signals to the beta cells, shown here in these dashed arrows, an increase in negative paracrine inhibitory tone, and a boatload of bad inflammatory signals. So all of these cell types seem to be abnormal, and they all could potentially impact on the beta cell. Okay, so what are we doing to study this further? So we're really lucky to have CF Foundation funding, and I haven't got any data for this yet, but we're going to be taking pancreas, or we are taking pancreas from donors with and without CF from CFRD. We're going to take frozen cells, and we're going to do single nuclear multiome analysis in collaboration with Kyle Galton. If you were at this session last year, he gave an awesome talk. So excited to work with him on this. And then we're going to be doing spatial molecular profiling on the right on tissue sections, where we can look at gene expression and put it into a two-dimensional map. So we can look not only at what the cells are making or not making in CF, but how the cells are talking to each other, and really try and get some actual molecular information to try and fill out this map that I've built for you today. So with that, I'd like to thank the patients and families that have donated their tissue, our funding, and my collaborators, and I'd be happy to take questions.
Thank you. Okay. Well, I'll go ahead and ask a question. I'm curious, do you think that the vasculature is different depending on the severity of the CFTR genotype?
Yeah, that's a really great question. I think, have enough of a sample size that we might be able to start to get at that now. My sense is that certainly the severity of the loss tracks with pancreatic exocrine insufficiency. I think where we see almost complete sort of fatty infiltration in the whole exocrine pancreas, that's where we see the most decrease. And I haven't tracked that back to genotype yet, but I wouldn't be surprised if it does track. Yeah.
Okay. Any other questions? Okay. Well, thank you, Rebecca. That was a great talk.
Yep.
All right. So our next speaker is Eddie James, and he'll be discussing the immune system and CFRD. Thank you.
Well, I appreciate the invitation and the chance to share our work. I have no relationships to disclose related to this presentation, other than the disclaimer that this is a relatively new project, and I'm relatively new to the CF world. And so I'm eager to be here and learn not only in this session but at this conference overall about CF and all the excellent work that's going on. So for our project, I come from a type 1 diabetes background. And so, my interest was piqued when I saw the Hart paper come out showing that CD3 positive T cells were increased in CF diabetes. And so what kind of formulated in my mind as I bounced ideas off of more knowledgeable people and as I thought about what I've learned in type 1 diabetes is, could there be an immune component to that disease? And kind of coming out of that, I really came up with three key questions that I wanted to answer as I thought about this disease and a possible immune component. One would be, is there immune dysregulation present in individuals with CFRD at a very broad level that could be detected, for example, in peripheral blood? Secondly, is there immune recognition of the beta cell antigens that might be playing a role in the etiology of CFRD? In other words, is there an autoimmune component to this disease? And thirdly, what are specific immune pathways that contribute to the development and progression of the disease? So kind of distilling those first 2 points down into actionable points about how the immune response might be dysregulated, and ways that we could bring tools from other autoimmune diseases to bear to possibly correct those abnormalities. And of course, there is evidence in the literature suggesting that there could be immune dysregulation in CF. And this is all kind of cold from the literature. There are multiple reports that neutrophil tracking to and from the lungs may play a role in the disease. And this is an increase in processes like netosis, which is extremely inflammatory and can be related not only to infection but to other sorts of inflammation. Secondly, there's been shown in a couple of reports now that there's a Th17 phenotype in peripheral blood that's correlated with poor lung function in individuals with CF. And of course, Th17, that's a cell subset that produces a cytokine called IL-17, which is one of the most inflammatory cytokines that's involved in early inflammation in a variety of settings. It can be protective for certain viruses, but it's a key pathway in autoimmunity. Thirdly, there's evidence of dysregulated IL-2 and IL-18 production in individuals with CF. And of course, IL-2 is the signature cytokine of T cells. And IL-8 is a more generalized signal of inflammation, and it's very important for programming T cells to participate in inflammatory responses. And finally, there's histological evidence of localized inflammation, including cytokines such as IL-1 beta, which again would lead to a milieu that would program T cells to be inflammatory. And so all this, according to my thinking, and I admit I may be biased, it lends the idea that there's a smoking gun, if you will, that there may be autoimmunity going on. So what evidence is there for autoimmunity in CFRD? And again, a lot of this is indirect, but there is an increased incidence of beta cell autoantibodies in people with CFRD as compared with the general population. Now, this is minor. If you look at a healthy control and measure the classic autoantibodies, you see basically about 2% of people in the general population that are autoantibody positive. If you look at individuals that have CFRD, the published reports vary. There are instances as low as 8% and as high as 33%. So this is very controversial. But if you do a statistical test, any of those studies will tell you that there is an elevation of beta cell autoantibodies in people with CFRD. Interestingly, there's also a higher incidence of high-risk HLA haplotypes in CFRD. And by high-risk HLA haplotypes, what I'm referring to is there are specific HLA DR and DQ haplotypes on the class 2 locus that are linked with autoimmune diabetes, especially type 1. And these are also elevated in people with CFRD. Thirdly, there's evidence that there are immune infiltrates in the islets of, and this is again from the heart paper that Rebecca talked about, of organ donors with CFRD. And for one such donor, CD4 positive and CD8 positive T cells were isolated and shown to secrete inflammatory cytokines. And these, fortunately, are available for our study. And so basically, T cells are there at the scene of the crime, at least in this particular individual, and we're working on getting other case studies. And they make all the sorts of bad cytokines that you would expect to be involved in an autoimmune response. So again, a smoking gun. And finally, CFRD individuals who are positive for autoantibodies, thinking epidemiologically, they have a higher risk for severe hypoglycemia and ketoacidosis. And so you could argue that there may be even possibly two forms of CFRD, one that has more of an autoimmune component that is more difficult to manage and to treat. And so if there is a subpopulation of CFRD that has an autoimmune component, it would be important to know about because that may have important implications about how the disease might be managed. And finally, going through the literature, I came across an interesting case series. This is a paper by Kim et al. In 2018 that highlighted 3 pediatric CF subjects that had clear evidence of beta cell autoimmunity. These 3 had unifying case features that included a pre-pubertal age and high insulin dose requirement for the onset, so very early onset and very high insulin requirements. 2 of the 3 had high fasting glucose and were positive for beta cell autoantibodies. So they're part of that atypical group that has these high titer antibodies. The third differed from the 2 in that they had normal fasting glucose and they were beta cell autoantibody negative at presentation. But this individual subsequently was retested as being positive for autoantibodies when they had a sudden increase in their insulin requirement. And so in this case, it almost seems like they had a more non-autoimmune type of CFRD that then basically developed a secondary autoimmune component that basically caused them to spike autoantibody positive and to have the sudden increase in insulin requirement. And so this creates a scenario is that there may be certain individuals where autoimmunity may be part of the early phase of their disease, and there are other individuals that may have other mechanisms which undermine their beta cell activity, which then stresses the beta cell, which is something that I could give a whole other talking about, that then actually can cause the formation of new epitopes and antigens and actually recruit an immune response. And so basically you can have autoimmunity that's secondary to compromised beta cell function. So with all these things in mind, we then went on to frame a project where we could begin to tractably address these questions. And so our objective then was to basically, assemble a study and a matching cohort, to identify key features of immune dysregulation that are present in individuals with CFRD, to perform epitope-specific assays to identify beta cell antigens and T cell lineages that are relevant to CFRD, and finally to perform transcriptional analysis and functional profiling of self-reactive T cells in individuals with CFRD. And our study design is as follows. So we've done a power calculation, and we believe that we'll get very meaningful results if we can get 28 subjects per group. And so we are in the process of recruiting 28 individuals with CFRD, 28 individuals with CF that don't have diabetes, and then 28 age and sex matched individuals with type 1 diabetes as a reference population, and finally 28 age and sex matched healthy controls. And for these, we intend to do assays that include measuring their islet autoantibodies, doing in-depth HLA typing, doing global immune phenotyping by flow cytometry, and then doing epitope-specific T cell assays and the transcriptional and functional profiling of T cells that I talked about. And I guess as a self-spoiler, we're about halfway through that recruitment right now. And so for our CF and CFRD cohorts, we have set very stringent inclusion criteria and exclusion criteria so that we can do this study in a meaningful and safe manner. And so for CF, we've defined, that is having a CFTR genotype abnormality or using the sweat chloride test criteria so that we can include people that have been tested both ways. CFRD is being diagnosed, defined using standard ADA criteria. So, for example, 2 hour oral glucose tolerance tests with a value greater than 200, and we're requiring insulin medication because we really want a group that is definitively insulin dependent, to use as that population. And then for CFRD without diabetes, what we're requiring is normal oral glucose tolerance tests within 365 days of their enrollment in the study. And so our goal is to really have people that we're sampling for this that very clearly have CFRD, that are very clearly CF without, And then our exclusion criteria are all related to safety and practicality. So we're not studying minors just because of restrictions with blood volumes. We're excluding pregnancy, corticosteroids because that messes up the T cells. And of course, we're not allowing active infection or illness, and safety criteria like BMI, FEV1, lung transplant, et cetera. And so here's the subjects that we have recruited so far. So we have 15 individuals that have CFRD. We have tracked their age, sex, and CF genotype, and they vary. All these are Caucasian, mainly because these are being recruited in Seattle, and that's a very Caucasian-dominant area. And we have ages ranging from 19 all the way up to the mid-50s, and so a fairly broad age range. We have mirrored that recruitment with a fairly well-matched CF cohort. Again, various CF genotypes, again, primarily white, a good mix of male and females. And here ages, again, running from 19 to this cohort so far is trending a little bit higher in age. We're going all the way up to 66. But the mean age for both cohorts is in the 30s, which we're reasonably happy with. And so, being thus far on the recruitment, we think it's great that we have 15 in each group that we're going to be able to compare these. And since we're halfway through, we're beginning to do what I would call an interim analysis. And so we went ahead and so we've been collecting DNA samples, both large and small aliquots of peripheral blood, and then things like plasma and serum, so that we can send for the various tests that we want to send for. And so we took the DNA and sent for HLA typing results. And the good news is that, first of all, 90% of the participants have at least one haplotype that can be studied using HLA tetramers and using type 1 diabetes-related epitopes that are well known and defined. And so basically that shows that for these 30 individuals, we're going to be able to study their T-cell responses using available tools, which is very good news. Interestingly, 50% of the study participants with CFRD had the susceptible HLA class 2 haplotypes, those that are affiliated with having risk of type 1 diabetes. 25% of the study participants with CF but no diabetes had susceptible haplotypes. So, we haven't done stats yet because we're only halfway through, but it's an interesting trend that we seem to have an enrichment of the risk HLA haplotypes in our CFRD group. We went ahead and sent for autoantibodies. I think most of this audience I don't know if this audience is acquainted with these testing or not, but there are 4 standard autoantibodies that are used as the criteria for having risk of type 1 diabetes. And so these include the proteins GAD65, islet antigen 2, insulin, and then a zinc transporter 8. So these are considered to be the classic autoantigens in type 1 diabetes. And if a person has just one of these autoantibodies, they're considered to be at risk in what's now called stage 1 of type 1 diabetes. If you have 2 of these autoantibodies and you're in the general population, you're considered to be guaranteed to eventually get type 1 diabetes, interestingly enough. And so if you look at our CFRD cohort, I have highlighted in bright yellow here those that tested positive for at least one of these. And so we have a single GAD positive, no IA2, 1 ZNT8, and then a whole host that have been positive for insulin autoantibodies. But there is one important caveat for these positive results, and that is that all of these were taken after the individuals had been exposed to exogenous insulin. And there's some controversy about that, and that these people are injecting themselves with insulin every day, and so that may in and of itself prime an antibody response. However, it is interesting that it is not inevitable, that these individuals take insulin every day and get autoantibodies, because you don't see yellow in all of these rows. And so as we subsequently parse through the remainder of our immune data, it will be interesting to ask, whether these individuals who have spiked insulin, positive have become insulin positive, not only because they see insulin every day, but there may be some sort of an underlying T-cell activity going on that is reinforcing that. And so, that's an interim hypothesis that we're going to be adding to this study, is to ask whether, becoming insulin autoantibody positive actually means something on the T-cell side. So now, moving to our CF group, we see a lot less yellow. And so again, we have 2 individuals that are GAD. We have nothing for IA2, insulin, or ZNT8. Again, with the caveat that these individuals have not been exposed to daily insulin injections. And so we can't speculate about whether any of these would spike positive for insulin autoantibodies if they, for some reason, started taking insulin. And so we can only be agnostic about that. But summing all that up, so we have 8 of our CFRD participants that have autoantibodies, 7 with a single insulin, 1 GAD, and 1 ZNT8. And certainly that triple positive individual is probably going to be a very interesting case to study. And then 2 of our 15 CF patients have autoantibodies. Both are single GAD positive. And it'll be interesting to see whether those 2 individuals go on to have increased evidence of glucose, lack of glucose homeostasis as they go on through the study. And so our next course of business is going to be to take this first half of our cohort and to perform the detailed immune cell profiling that we've talked about. And again, this is a very busy slide that I don't expect you to memorize the details of, but to make a long story short, we have devised a panel with 35 immunological markers that are going to be able to enable us to do a very broad immune phenotyping of these individuals to look for a lack of balance in their immune repertoire and to probe for abnormalities. And this is a panel that's based out of a published study that came out of BRI that actually investigated people with Down syndrome who are known to have a very elevated risk of developing various autoimmune diseases, and compare them to type 1 diabetes subjects and controls. And what that study revealed is some very interesting phenomena, such as accelerated immune aging in that group. The analysis method that paper used that we're going to steal, because why reinvent the wheel when you can steal from the best, is an approach called IMPACT-D. So it's iterative machine-assisted analysis of cytometry data, which is kind of a cool little acronym. And what this method allows one to do is to take very complex flow cytometry data and to analyze it in a completely unbiased way. And so what this does is it gives you FDR-corrected discovery of a variety, and that's very important because when you have 35 different parameters and you're looking at both percentages and mean fluorescence intensities and dual and triple and quadruple expression, the multiple testing correction for that is enormous. But this parses through that in the most efficient way possible. allowing you to do very efficient discovery. Yeah, and so if you can start your discovery on key populations, and so highlighted here are things like B, NKT, CD4, and CD8 positive T cells. And then you build beneath them basically different node and subnode populations, and you're able to build trees. Yeah, it uses either user-defined thresholds or automated gating, but we're probably going to do user-defined thresholds because I don't do automatic gating. Yeah, and this then allows us to build some models. And what this, I'm not going to bore you with all the technical details because, first of all, I don't fully understand them. Yeah. But what this allows you to do is basically you're allowed, you can look at root nodes and then intermediate nodes and terminal nodes to basically identify which specific immune populations are being perturbed. And this slide here shows just a couple of examples. I'll see if I can steal the arrow here. And so if you look between these groups and ask, are the naive T cells different? You see nothing, which I guess is shocking. But so naive T cells can be a node. But if you then pass through the other markers and ask, okay, on naive T cells, are there any features that are different? Then this impact D approach was very quickly able to discover that, okay, if you have Down syndrome, you have this profound deficit in your CD39 expression on your naive cells. And again, it would take a person a lifetime to permutate through all these possibilities manually, but Impact D does this by itself, and so we can very quickly winnow in on basically main populations and subpopulations of T cells and B cells and NKT cells, and find signals where things appear to be dysregulated, and then parse through them to build models and draw meaning. And so we're very excited to take this unbiased and rigorous big data approach to parse through our data and figure out how the immune system may be perturbed in CFRD as compared to CF and type 1N controls. And then we'll move on to do what my lab actually does best, and that is antigen-specific T cell assays. And so one thing that my group has done over the years is we've become leading experts in what T cells see in autoimmune diabetes. And here's a list here of all of the epitope-specific reagents that we have developed and have available for our use. You'll see some hopefully familiar protein names here like proinsulin and GAD65 and IA2 and ZNT8 matching the antibodies. But then there's a whole other very interesting proteins that are recognized by T cells, including even some surprising candidates like cell cycle and nuclear envelope proteins. And these can be seen in their modified forms or unmodified forms. But basically what we have here is basically a grid to really ask in a very deep way what T cells are seeing in these individuals. And we'll be applying these assays both to peripheral blood samples and to the cell lines that we've gotten from the pancreas to basically figure out, is there an antigen-specific signature in these diseases? And we think that'll tell us a lot about what the underlying root cause of CFRD may be in these various types of patients. And then finally, we're going to use all of this as a means of doing detailed single cell analysis of cells from the peripheral blood of these patients. And so what we can do here, again, is we have established at our institute a very efficient pipeline where we can sort single cells of interest onto a plate. We can then also record the surface marker expression to index them based on the immune populations that we identify through Impact D. And then we can do basically RNA sequencing analysis at a whole genome level. We can also build up their TCR sequences, and then we can cluster their cells and basically look for different subsets of cells that are disease specific. Comparing naive, regulatory, Th17 sorts of phenotypes. This grid here is from an RA project, but we'll build up our own for this. And what are our expected outcomes? What we think is that we're going to see that immune phenotyping will reveal immune features that are shared by all subjects with CF and others that are unique to CFRD. We expect that we will discover key beta cell epitopes that are present at elevated frequencies in subjects with CFRD as compared to controls. And we expect to show that T cell responses in individuals with CFRD have distinguishing functional and transcriptional features. And so with that, I will move to my acknowledgement slide here and just thank the members of my own lab that have helped us build these systems, the various core laboratory groups at BRI that make this sort of research possible, including our tetramer lab that makes all of our reagents, our patient registries that help us recruit the type 1 diabetes and reference populations, and then big kudos to the group at the University of Washington led by Kathleen Ramos and Eric Morrell, who have done and are continuing to do all the recruiting that we need to build this study. To Sally Kent, who's an expert in isolating T cells from the pancreas, who's provided all the cell lines that we're going to be using for this. Bernard Core, whose group developed Impact D, and then other collaborators through the years that have helped us gain an understanding for autoimmunity in the pancreas and how T cells behave. And of course, many thanks to our funding that they're listed on the bottom, especially the CFF that has funded this as a new project starting at near the close of last year. And with that, I'll take any questions that have come to your minds. Thank you.
Hi. I think this is really important work, and I'm glad to see you doing it and that you're doing it so comprehensively. You're going to be up against some skepticism, maybe not so much in your expected outcomes as in the initial correlations with type 1 diabetes. I think that the prevailing thoughts, at least right now, are if 1 in 300 people in the general population has type 1 diabetes, 1 in 300 people with CF are probably going to have type 1 diabetes independent of their CF. And when I look at your data and your subjects, I don't put a lot of stock in insulin autoantibodies in somebody on insulin therapy. I think you have one subject who clearly has type 1 diabetes, and I think of your non-diabetic subjects, isolated GAD is a little controversial. So I'm not convinced about that link with type 1, but we don't have a lot of data, and you're going to produce it. So kudos to you for doing it.
And I'll acknowledge that all of those thoughts are fair. And the good news is that, I mean, our project isn't completely dependent upon there being a direct parallel between the two diseases.
Hi, really interesting data. Catherine Cutney from Case Western. I probably missed it, but are your CF patients without diabetes all pancreatic exocrine insufficient? Scott likes that question.
To be honest, I'll have to check with our clinicians on that. I don't know.
Yeah, no problem. I just wonder, like, just from everything you know about diabetes in general, is it possible that having the exocrine inflammatory environment is a prerequisite to developing the autoimmunity? Like, would being surrounded by all that inflammation somehow trigger the autoimmunity against the beta cells? What do you think of that hypothesis?
It's a reasonable model, and certainly there's a lot of inflammation that would come out of the signals from excellent exocrine inflammation wouldn't do anything good. We'll put it that way.
Thanks for the great talk. Bryn Marks from Children's Hospital, Philadelphia. We know in type 1 diabetes that the timing of the pancreatic autoantibodies, they can appear and disappear at varying times. And to the point of Tody's concern about looking for insulin autoantibodies in people on insulin, have you thought about looking sooner, immediately at diagnosis or at various time points within the diagnosis of CFRD?
That would be the preferred methodology, and I'm coordinating with our clinicians to go back as far in time as we can to see what antibody tests were ordered early on. And but I completely agree that autoantibody test results that by far precede first insulin use would be much more meaningful. Yeah, but not always routine to do that.
I think you've got in hand something that was I really love how you showed us all the CFTR genotypes for the people in your two cohorts. And Dr. Hull did it too. So I think that's really important for everybody to tell who is feeding into this really important study, right? Because you're going to compare CFRD and no CFRD. And it's really important to know whether they have genotypes that confer pancreatic insufficiency or not. Mm-hmm. So I noticed that GAD positive CF patient has a CFTR mutation called S292F, which confers pancreatic sufficiency. And so that makes just because you were able to share the data, it makes me look at that and say, I wonder if they are a pancreatic sufficient patient who has autoimmune positive diabetes, and that's many people would call that type 1 until proven otherwise. In the control population, you've got a bunch of, adults who don't have diabetes, and many of those people have, survived to their 40s and 50s in the last 4 to 5 decades. And I'm noticing also that about half of those people have mutations that confer pancreatic sufficiency. So I think that would be really, really helpful to look at that carefully as you define these two groups. thank you for sharing all of that.
Well, yeah, thanks for the input. And again, I'm a relative newbie here, but yeah, I've backed off from standing too high on that hill. But it has been my hope since we have started this study to be able to draw some inferences about CFTR genotype and how it plays into this system. But I don't. We won't know until we recruit whether we're powered to ask any questions or not. But hopefully, yes.
Thank you. And our next speaker is Katie Larson Ode from University of Iowa. And I'll do this for you. I'm good to go.
Okay. Hello. I am very excited to talk to you guys about our study. And I'm speaking for the PI group, which includes Dr. Christine Chan from Children's in Colorado, Dr. Mira Mohit at the University of Minnesota, and Dr. Andrea Granados, who was at Washington University St. Louis while she was working on the study, and now is at Nicholas Children's Hospital. So I just wanted to make sure I acknowledged all my co-Is before I got going. So this is my disclosure slide. I have disclosures. I have consulted for Vertex, which is relevant to the study, at least somewhat. And then I have funding from the CF Foundation, from which I'm incredibly grateful. And then I have some other funding as well. And the University of Iowa considers the fact that I edit for up to date to be a conflict of interest, so it's on there. This was originally titled Current Studies in CFRD. So this is, I just went to clinical trials and pulled all the active studies that are relevant to cystic fibrosis-related diabetes that are currently available in clinicaltrials.gov. So this is pretty cool, and there's a lot of stuff going on here that I'm desperately hoping that these studies will be completed and we'll have the data. When I was looking through clinicaltrials.gov, I was so depressed by how many of the CF trials have been withdrawn that have been on there historically. So hopefully these will all fly and we'll get all this data. Obviously, I'm going to talk about the one I know stuff about today. So why did we do our project? Probably the good place to start from is that all the ways we define diabetes, and you may notice in the previous talks, we use ADA criteria, which are actually based on type 2 diabetes. And as we've heard very clearly for all the talks that preceded me, obviously CFRD is not type 2. So we don't have definitions of diabetes based on people with CF currently. And so the CF Center in Milan found that they could use very elegant age and sex-dependent measures derived mathematically from OGTT to predict beta cell decline, because that's what we're looking for. We've had such a good intro, like everybody knows everything already, so I don't have to explain anything. So like we're looking for beta cell decline because the beta cell lack of production of insulin is the problem, as we already learned. So that's what we're trying to predict, right? That's how we're going to try to figure out if people have CFRD. So the goal of this study was to develop age and sex-dependent reference ranges for frequently sampled OGTT-derived estimates of beta cell function. Sorry, I'm going to have to say that a whole bunch of times. And they're envisioned kind of as growth charts. We know that age is actually the strongest risk factor for diabetes in people with CF, and that it's very different in males and females. So we know we have to adjust for both of those things. So this is the person whose fault this whole thing is, more or less, that and Tony Moran. So this is Alberto Battazetti. He is an endocrinologist in Italy who specializes in CFRD. And he actually did this study in Italy, more or less. And he approached us to see if we would do something similar, because obviously these would be very different populations, and if you find similar things, you have reproduced it, right? So this is he started before us. This is the publication of the study that he did. So for our study, it's a little bit different than what he was doing, but it's related. So our first aim was looking for these age and sex dependent reference values. And goal was to get 475 visits in our study. And our goal was to have a diverse population. You're going to find out how well I succeeded. And then we were going to look at glucose levels, insulin levels, C-peptide levels, and then measures that are mathematically calculated, including beta cell sensitivity to glucose, oral glucose insulin sensitivity. And we're hypothesizing that those are going to fall along these kind of growth curves dependent on age and sex. And then for our second aim, we were going to try to relate those basically to CF relevant outcomes, including FEV1, BMI Z-score, and the number of pulmonary exacerbations over the past 12 months. And we figured those would also be age and sex dependent. So because we were going to do a relatively big study, we're like, we have to get as much data out of this thing as we can. So we put together a bunch of sub-studies, and each of us was a leader of one of the sub-studies. So Dr. Granados is a leader for the DEXA sub-study. And her hypothesis was that if you had less lean body mass, so less muscle tissue, then those would be the people who would also have the worst beta cell function markers, and that would also be, of course, age and sex related. And then I came up with the islet hormone and incretin part, and obviously I assume those would vary by age, which based on the data we just saw is probably true even if we don't find it in our study. And then I had thought, I just basically made this up, that the beta cell glucose sensitivity would correlate with pancreatic polypeptide and GIP, but not with glucagon and GLP-1 in the serum. So I don't have any idea if that's true. And then Dr. Mohit was looking at hypoglycemia, and we figured that reduced beta cell glucose sensitivity would be the people that would have more hypoglycemia. And also that we would be able to look and find some inappropriate catecholamine and glucagon responses that were more severe in the people who are having hypoglycemia. And then Dr. Chan, of course, led the CGM part of the study, which was looking at glucose in the free living environment measured by CGM. And then we were going to see how well that did or did not correlate with what we were looking at in the OGTT. And those are the specific outcomes that she's looking at there. So we have 4 sites. University of Iowa recruited both pediatric and adult subjects. University of Colorado recruited pediatric patients. Minnesota recruited adult patients, and WashU recruited peds and adults. And we actually were very open because we were trying to get as many people as possible, and we wanted to be representative of the population. So it was really, all we wanted was you didn't have diabetes right now, and you were at least 6 years of age, and you weren't sick or newly on steroids. And if you were on a corrector, you'd been on it stably for a while and hadn't been newly placed on it. Because we knew when we put this study together, this was before, the big event, but we knew people would be on correctors during the study. So we knew if we excluded that, we wouldn't have anybody in our study. So we did a frequently sampled oral glucose tolerance test with 8 time points, which are listed there, standard glucose dose, and then everybody who was in the study got glucose, insulin, C-peptide measured at all those time points. Then we did an exam, because of course I'm a pediatric endocrinologist and I like to make sure that I know how big people are. And we did Tanner stage people, important to know if they're in puberty. And then they did a hypoglycemia questionnaire during the OGTT. We reviewed their medications carefully. We sent them home with a hypoglycemic questionnaire for home. And we did extensive chart review for CF relevant clinical outcome measures. And the sub-studies were not done in all the subjects. DEXA was available to people who were interested at Iowa and Colorado. CGM was available at all sites, but not everybody did it. And then some of the participants at every site were eligible to have the incretin and islet hormones done. And then we measured A1C at Iowa, Colorado, and WashU. So this is our study timeline, and a bunch of stuff happened between when the study started and now. So our IRB was approved in March of 2019. The first visit happened in July of 2019 because it takes a long time to get a large 4-site study up and running, and there's a lot of logistics that I didn't fully appreciate before I started doing this. And then, of course, this happened, which everybody here knows about. And then this happened. It was great. And then this happened, so Dr. Granados moved, so we had to close the WashU site. And then we also found that our insulin assay may or may not have had issues. So we had our last study visits at the end of July of this year. And so we are still cleaning and getting the database ready. We don't have everything ready to go, unfortunately. We did have opportunities that arose because of those things that happened and did result in a couple of things that we looked at. So Dr. Granados had the opportunity to compare some of the DEXA scans from people who, before and after they started on highly effective modulator therapy. And so we were able to publish this. And what we found, very short version, is that they gained weight, as you guys all who have ever exposed to Trikafta know. They had increase in their fat mass, but not their lean mass, which is, I think, really interesting. We saw increased markers of insulin secretion. We use C-peptide and not insulin. And then C-peptide-derived markers of insulin resistance were also increased. And then that was a smaller group. In a larger group of the patients, because not everybody who went on Trikafta had a DEXA. In our bigger group, we looked at a little bit more intensive investigation of beta cell function to try to figure out if, that improvement in insulin secretion was like meaningful. And basically what we found is, it looked like they were making more insulin, but they're also more insulin resistant, and it didn't come out to improving anything. We don't have any of the final results yet, but I'm going to share with you what I have. I pulled this data Monday night and Tuesday. So this is really fresh, and my co-Is haven't even seen it. So what do we have for the specific aims of the primary study? Well, we recruited 314 unique subjects, not 475, but they did complete 482 visits. So I just want to this is pretty good, right? Pretty good? It's a good number of people, especially for 4 centers. And so, as we all know, you don't always get all the data you want when you have a complex study. So of the 314 unique subjects, 294 of them had at least one visit that had analyzable OGTT data. And then of the 482 visits we had, 435 of those had at least some OGTT data. People were eligible to do the study yearly while the study was involved. It was envisioned mostly cross-sectionally, haha, envision. But people were eligible to repeat it yearly while the study was ongoing. So we had most of our people who were participating were only in for one visit because we were continually enrolling throughout the study. And then we had a lot of people who did multiple visits. So, which should be pretty neat because we're going to have at least some ability to do some longitudinal analysis. So, as I mentioned, we don't have our full data analysis, so I don't have anything from the answer to AIM-1 for you, especially since we have to redo. We're actually have redone already most of our insulin samples, and we're going to complete that, and then we need to pull that in and clean the data to make sure that our insulin data is good before we try to do anything with it. So, and since everything is dependent on the insulin data, we have to have good insulin data. So, and that was another good thing that came from the intermittent analyses that we did is we found when we started doing the math for the looking at insulin sensitivity, Dr. Mohit figured out that everything was coming out funny on the calculations and that there was probably a problem. So, which was good. So, and then AIM-2, the problem with AIM-2 is Alex-Tez-Iva. So 45% of our participants were on this at their first visit. And of course, as you go forward with the visit numbers, it's higher percentage. And so, of course, that's going to affect BMI. That's going to affect FEV1. And nobody's getting admitted for pulmonary exacerbations, right? So our exacerbations in 12 months is probably not going to be high enough to be statistically meaningful between the groups. So I don't know if we're going to look at it, but I don't know what we're going to get in the context of highly effective therapy. So for our sub-studies, we had 164 people who consented to CGM, and 130 of them have at least some data. We had 97 participants who had DEXA. Whoops, and I didn't put it on another bullet for some reason. 214 people had A1Cs. So here is our table 1. And unfortunately, there's a lot of stuff on there, but let me see if I can make where'd the mouse go? Trackpad. Oh, trackpad. Okay, sorry, trackpad. All right, so I have it set up. I'm just going to orient you a little bit because there's a lot of data here. So this is everybody who had a visit even if they didn't have OGTT data available. These are the people who had OGTT data available. And then I split it out by age ranges because this is what we're really interested in what was happening by age. These are the same age groups that Dr. Badazetti uses, except I made one older one because this group is different. So what you can see here is we actually did pretty good. Our median age is 18, which means basically half our subjects are adults and half our subjects are peds, which is great. And so this is mean, and then first and third quartile is how this data is shown. And then we had majority male in almost all of our age groups and in the overall population. As you would expect, most of our patients were pancreatic insufficient. And also most of our patients most of our patients had at least one copy of F508del, and we actually had a pretty high percentage of people who were homozygous for F508del. We had about 10%, this is very representative, I think. About 10% had no copies of F508del. So, and then our diversity was not as good as I was hoping. We had 5% of people were identified as Hispanic, and only 1% of our patients identified as Black, 2% as Native American, and then a few people declined. So we were a very, very, very white population, which is not ideal. And then the main thing about all these age things is actually it's pretty consistent as far as these things go, except for when you get our upper end of our age range, and especially people over 40, so like people my age. These a lot. The male-female balance is even more discrepant here, and that has to do with mortality and gender in CF, I think, because we know women have a higher mortality. And then obviously, where did my arrow go? And then obviously the pancreatic sufficient goes way up, right? And then there was one person in this group that we don't know if they're pancreatic sufficient or not because they didn't get tested. Somehow they kind of just got by that somehow. And of course, the older population is even less diverse than our younger population is. The only good thing about diversity is what you do see is the people, our minoritized populations who were in our study, they all have data. So they were all really good participants in our study, and we have data on all of them. So that's the one good thing. We didn't lose any of our minoritized population. So that was good, but we really needed better enrollment than we had for that. So these are the risk factors for CFRD that we have in our population. So this is just from the first visit. And so what you see here is this is all the different correctors. And I use the brand names here just to make it easier to tell who was on ivacaftor for G551D, before they went on Trikafta. So they are on Kalydeco for the indication. So 54% of our subjects, I mentioned it was like 43 that were on ETI, but it's actually 54% of our subjects were on some form of what we would consider highly effective modulator therapy at their first visit. And of course that goes up over time. And then family history of type 2 is a strong risk factor for the presence of CFRD. And then you can see that 54% of our participants also had a positive family history for diabetes, and for 40% of those, it was type 2 or CFRD, and maybe type 1 is really relevant, as we just heard. So anyway, we have a lot of participants who had a strong family history of diabetes. And so here's our BMI information for our study. And really the takeaway for this is our pediatric patients really were doing fairly well from a BMI perspective. Our adults are not. What you can see here is that 40% of our female participants had a BMI under the goal, and 32% of the males did, but then we still have 31% of our adults who are overweight or obese. So we really have, and this is, I think, consistent with what everybody else is seeing, but we definitely have that's there. And so that would be expected to impact probably insulin resistance. So we don't have final outcome, but I can show you a bit of our glucose data. So this is the glycemia. So we had tried to exclude people who had CFRD, but we still ended up with people with CFRD range, blood sugars. And what I'm showing here is the time points where they met the criteria. And so we did have a couple people who had CFRD range fasting blood sugar without CFRD range any other blood sugar. So I don't know if that's what happened with those people. We'll have to dig into them a little bit more. But, and then this is comparing those percentages for all the visits versus just the first visit. And you can see it's pretty stable, which is good. And then, so I mentioned that already. So although we had attempted to exclude CFRD, we still had people with CFRD. Most of our participants had some sort of glycemic abnormality. We had very few people who were fully normally glucose tolerant. And then here's our A1C data. Of those we were able to get an A1C on, almost exactly half of them were over 5.5 and half of them were under, which is actually pretty consistent with the Toronto data. That would indicate 5.5 is a reasonable cutoff for like the likelihood of having an abnormal OGTT. So hypoglycemia, we had a lot of hypoglycemia. We elected to look specifically at two different cutoffs. Less than 55 is really concerning for hypoglycemia. 70 is an alert value and may not necessarily mean, it might not necessarily be abnormal. We don't know for sure. But what you can see here is that, is that we had a pretty substantial rate of people who had hypoglycemia during OGTT. So we had 21% of people who got less than 55 during their OGTT, including a person at Colorado who started less than 55. And then, and this is the visit 1, and this is the same thing for visit 2. So you can see it's very stable. And what you can see is really the low blood sugars really happen mostly after 120 minutes for the vast majority of the people. And most of the time in real life, clinically, we're stopping at 120. So, but when you go past 120, you're seeing a lot of people dropping low. And we don't know if that's normal or not or what that means, but we're going to look into it. And then this is something we did that was kind of, I think, useful to have, is we asked the patients if they were, the participants, if they were having symptoms of lows when this was happening. And we had 30% of our 28 to 30% of our participants actually had at least one time when they felt like they were hypoglycemic. And then we had a substantial, not huge, but real number of people who had more than one. And then we actually had 2 to 3% of our subjects that had to stop because they were low. And we didn't make them stop unless they were really, really low or really, really symptomatic. If they felt fine and they were like not in the 40s, we let them keep going. So, because we were trying to get those catecholamines and glucagon, right? So we needed to not treat people right away. So next steps for our study, as I mentioned, we're going to rerun all the insulin levels. So we have paused analysis until we get those updated levels. We're currently cleaning the database. We have a biostatistician, to help us with our modeling. And once we have the updated insulin levels, we're going to get going with that. And as I mentioned with AIM-2, we're going to do the analysis, but we're concerned that it may not be as meaningful with the advent of ETI. FEV1 and BMI may not correlate with beta cell function. I don't know. And it's at least 45% of our population. So, and I think the sub-studies will be interesting no matter what. So I'm excited to see what we get out there. And we have banked all these samples. They're in my -80 freezers at the University of Iowa. We're happy to collaborate with people. So if people have cool ideas for what we can do with these samples, we're happy to chat. So, and then I have so many people to thank, just like everybody else does. First, thank you to Cystic Fibrosis Foundation, who gave me money to do this. And then to all of the fantastic people that helped us at all of our sites. And I think I forgot people on the list. I tried to add people, but I might have forgot people. But thank you all. A bunch of the people who helped me are sitting in the audience right now. And I'm happy to take questions.
Thank you, Katie. Any questions in the room?
I think people need to escape. Looks like people have to go somewhere. Did we run over?
No.
No? Okay.
Yep. This is scheduled until 4, plenary's at 4:30.
Okay. No questions?
So you probably collected this. I didn't hear you say anything about overnight 2 feeds. And I guess you've got that information. So
Yeah.
I'm looking forward to hearing about that.
We have it in it's collected from their, it should be collected from their chart review. It's one of the there's a lot of stuff that we made the poor coordinators pull from their medical record and put into the database for us. A lot of stuff in there. Like if they're on steroids, what they're on, how long they've been on, because we took people on chronic steroids, just not people on acute steroids, and all their different meds, and when they went on correctors and off correctors and initiation date of correctors. And yeah, we collected a lot of all that stuff, but we didn't exclude people. So good question though, because that does make a difference.
I actually have a question for you, Katie.
Uh-huh.
Did you notice a difference with body composition and glucose tolerance, particularly following ETI?
Did we look? No. The glucose tolerance did not change in any of our pre-post data. We're going to probably stare at that again, but once we have the new insulin levels. But really, we did not see any change in glucose area under the curve or any of the time points. We did see a slight improvement in A1C, which mirrors some of data from other groups, but we did not see any improvement in actual blood sugar values.
That was amazing. I'm excited to see the insulin data when you have it.
I know. Me too.
So based on what you said, you probably don't know the answer to this yet, but
You can
I
ask
want
me.
to ask you anyway. So for the glucose tolerance categories, but really also for the hypoglycemia, it's really striking that you're seeing very similar proportions between the two different study visit types.
Yeah, I know.
Are they the same people, or do you know?
Are they low?
So-
No, I haven't been able to pull that yet. Yes, we
Yeah.
were chatting. We actually had a little like work meeting over lunch right before this session, and the other PIs were asking me about this because we were talking to the biostatistician. But yeah, I have not. I think from staring at the database that a lot of those people who had multiple sets who were low were the same people, but I don't actually know that for sure because I haven't it's just for me staring at stuff for hours and hours, not for me actually pulling any data in a proper fashion. So I think so, but I will have to check. We are going to look at that. Don't worry.
Okay, cool. Thank you.
Thank you so much for the research you're doing. I was curious if any of the centers that you're collaborating with are pulling fecal elastase, and if you're going to track any patients who may have updated diagnosis of pancreatic
Did we
insufficiency or-
pull fecal elastase? Do you guys? I can't remember. I think we did pull fecal elastase. I did in my. I don't remember. I could. Do you want me to get my red cap? I can show. If you want to come harass me in a second, I can pull my red cap and look.
We can do that part in the panel
Yeah,
discussion.
we can do that later. I have my computer with. I can totally pull it up and try to figure it out. I can't remember. I should, but I don't know. Okay.
Okay.
Any other questions?
Well, it looks like we probably have about 10 minutes for a panel discussion. There are a few questions that were asked after our speakers left the podium. So we'll bring all of them back and try to run through some of these. And please feel free to ask questions at the mic if you have any lingering questions from earlier presentations.
You're asking the exact same thing.
Also, I want to go ahead and put up this slide because the 7th edition for the CFRD manual came out and it's available. And these are the options you can use to get the manual.
Okay. Well, shall we do one of these over here?
Yeah. I think-
So, okay, so we had a question asked from over the internet, and it was, do you know of any research, and well, this is gonna go to everybody. Do you know of any research being done to differentiate CFRD versus type 2 diabetes in individuals with CF who, especially those who are overweight or obese or on modulators?
Everybody's looking at me. So, concerning the fact that I obsessively stalk all of the clinical trials that are going on in CFRD, I don't believe anybody is specifically looking at that. And I don't actually know how you would tell other than because I will tell you, the Toronto group has pretty good data on insulin resistance. It absolutely increases with age. So people with CF absolutely can be insulin resistant. Historically, they weren't. That was probably because they were malnourished. I would say with in our study, we did see increased insulin resistance with increased weight gain. And then, of course, The other way you would tell somebody has type 2 is you would gene test them, but somebody else has done that and shown that it absolutely, people with CFRD have have evidence of type 2 susceptibility genes. So that would be a really challenging, I'm not sure how you would decide that it was type 2 and not CFRD, to be perfectly honest with you. And the thing is, it's the same kind of question as when you have somebody with type 1 who's insulin resistant, they still have type 1. Type 1 is an autoimmune disease, right? So if you have type 1, it doesn't matter how much insulin resistance you have and how much treating insulin resistance will make your diabetes better. You still have type 1. You might have type 2 too. I don't know. But I don't, so CFRD, it has a true pathology. We saw that. That's very specific. That's probably caused by the exocrine pancreas disease. So I would say if you have CF and you have diabetes and you don't have raging antibodies, or maybe if you do, you still have CFRD, and then you probably also have a bit of type 2 phenotype. Now, do I have a young man that I take care of who is Native American descent, who is, everybody in his family has type 2 and is really overweight? And did I just put him on metformin? Yes, I did. Because he really looks like he has type 2, and he has very atypical CF, and he's pancreatic sufficient. But I don't know how you would design the trial to do that. I think everybody wants to know, but I don't know how you would define type 2 in a way that you could exclude, definitively separate them enough to study them in people who had CF. You can talk about how people are insulin resistant and whether or not we should use medications that treat insulin resistance. That people are doing. Several of the studies I put up there on the list of studies are trying. People have been trying very hard to answer the question of whether we should use medications that treat type 2 in people with CFRD, but whether we could distinguish CFRD from type 2 well enough to study that, I don't think we can do it, to be perfectly honest. There's another question.
Go right ahead.
Are you talking about like the use of weight loss medications? Oh, lifestyle.
Everybody heard the question?
Yeah, sorry for the question. The question was for people who have weight gain with, is that okay if I just, you guys are not, these guys aren't clinicians, so. For people with weight gain with CF, how do you recommend weight loss interventions? And probably for people with diabetes too who have CF, right? So I, as a mostly adolescent medicine provider, am very careful how I talk to people about weight loss, because eating disorders are serious, they are deadly, and our CF patients are at higher risk for eating disorders than people who don't have CF. That being said, I have been picking on people about CF, about healthy eating, since the day I was trained by Tony Moran to be a CF endocrinologist. So have I always told my patients they can't drink pop? Have I always told my patients that they can't eat candy no matter what the CF team is telling them they can do? Have I always told my patients I need them to eat fresh fruits and vegetables and that I need them to stay away from processed food? Always. So when I talk to people, I never tell anybody to lose weight, even my 250-pound kids with type 2. I just don't talk about it that way because it ends up being very counterproductive. Most people who are overweight have a ton of medical trauma from doctors who won't take care of them because they're heavy. So when you're talking to people about losing weight, you have to be really careful. But do I do things to try to help? Yeah. And I actually have an adult who's really been struggling, and she's just been gaining weight like crazy on ETI, and she does spin class all the time, and she's working really hard. She's a super healthy diet, and her weight just keeps going up and up and up. Did I just put her on Ozempic? Yeah, I did. Did I email the pharmacy to make sure she could keep getting it while I was here at this meeting? Absolutely. So, we do what we have to do to try to help our patients get better. I wish we had more data on whether these things really work and are safe in CF. We just don't have. We have a little bit of data now, but not as much as we could use. But people are trying to get more. But when I talk to other endocrinologists who do CF, especially if they do adults, most people are using some of these medications and are working with people to lose weight. And I know that our nutrition friends are going to come out with some new nutrition guidelines to try to help address these issues. It's kind of funny with the whole thing with the ETI and the weight gain because it could be that ETI is obesogenic, but also, you know, when you're catabolic and you're sick, you're always less heavy than you would have been. And when I look at the BMI on my kids who are really heavy, who have CF now, it typically matches what their BMI was when they were 2. It's not usually higher, and it doesn't usually get higher, and it usually matches their parents' BMI. So you have to, our general population is over 30% overweight and obese. So I don't know if we actually have a problem or we're just making people American-style healthy is the only thing I would throw out when we're talking about all this stuff. So, healthy in quotes. So that was a long answer to a short question, sorry.
And I just would like to echo, I think that a lot of our adult colleagues, I'm a pediatric endocrinologist, so I don't encounter obesity as much in my CF population, but my adult colleagues do, and a lot of them are employing a lot of the type 2 medications for this population. I think the reality is that as time moves forward and people are on highly effective modulator therapies for longer, this line is going to be blurred between CFRD and type 2 diabetes. And there will be a crossroads and maybe a hybrid of how we're treating how we've traditionally treated CFRD and looking more like a type 2 picture.
I'd love to jump in too. I know you have a question, but
Yeah, all of us CF
Yeah.
endoclinicians
So
have really big opinions on this stuff.
So at some point, so people with CF have abnormal insulin secretion and lots of other hormone abnormalities, as well as many other non-endocrine abnormalities. But so if you have an obese person who has CFRD, does that suddenly mean that all of the type 2 diabetes medicines are going to work and be appropriate? The answer is no. But you know, with appropriate data, then it may turn out that there are things that would be helpful in this population. And so that's some of the exciting studies that are going on to look at some of those things. But I would caution people about just jumping in and using medicines that might be approved for use in diabetes in adults or even in children, and think about whether enrolling them in one of our exciting studies would be a better way to get the information as well as find out if that's a useful endeavor. But at the end of the day, I sort of always say it doesn't really matter what you call it as long as you decide what to do.
Yeah, I mean, I guess that a good example was from the physician in Grand Rounds this morning when they were talking about the guy with heart failure and got put on standard of care treatment for heart failure, which included a hardcore sodium restriction. And then he got very, very, very sick because people with CF have to have sodium, right? So we have to remember that our pathophysiology of our CF is still there. And even with highly effective corrector therapy, it's not the same as if you never had CF. So we have to keep that in mind, which is a really, really good point. We can't assume the pathophysiology is the same. And endocrinologists love pathophysiology, right? So we like to know why things are happening.
So I'm not sure if this is the right place to ask, but because we're talking about weight, which is controversial, and type 2 medications, which is controversial in the CFRD world, I'm an adult endocrinologist with interest in diabetes. I treat CF as well as all other types of diabetes. There's a lot of awareness about weight loss medications, so a lot of patients will push and ask for that. I have actually seen very good results in non-CF patients, non-CF diabetes patients, because with the weight loss, I understand the stigma with the weight and that, but I do see a lot of improvement in quality of life with the weight loss. Not just diabetes control, but they can move better, they can feel better, they can go out better.
I didn't mean to imply that we shouldn't try getting people to lose weight. I just meant how you talk to it about it.
Yeah.
I mean, I've had so many patients, you send them to the surgeon says, well, you can come back for an operation when you've lost weight. And they send them to me and say, make them lose weight in 3 months. And I'm like, right. So that's where I'm coming from. It's not that losing weight is good for people. We know that. Everybody knows that. It's just that I was mostly talking about how you talk about it.
Yeah, sure. But my question was a little different. So when I treat type 2 diabetes, when we treat type 2 diabetes, we are not monitoring their nutritional values. Like I do check vitamin B12 and D here and there, especially if they're metformin, but we are not having a whole panel like we do for CF patients. But I have few patients who I have put on GLP-1s, and they had like very, very educated patients follow up. They had amazing response, doing really, really well, lost weight. but their appetite has decreased. Their nutritional markers that they have been checking are all stable, but that does make my pulmonary and nutritional colleagues like really, really uncomfortable. And we have had like lots of discussions. But when I go to other diabetes, if they eat like one meal a day and if their all parameters look healthy and they look healthy, we don't really say that you have to have 3 meals. But in the CF world, I don't know how to address that, even though if we have more markers than other general population.
I don't think we know the answer to that with corrector therapy. Before corrector therapy, they couldn't skip meals, right?
And I think one of the words that I'm hearing come out of the nutrition guidelines is going to be individualize.
So,
Thank you. This is Mahesh from University of Wisconsin. This is a question for the first speaker, to be very specific. So in patients with CF, have there been reports, or like in the pancreas specimen, has there been reports of ductal transdifferentiation into islet cell, then acidioblastosis has been seen in addition to acinar cells, or it is more of acinar cell transdifferentiation? If you could comment on that.
This question is they was about whether the acinar cells transdifferentiation into the new generation acinar cells.
Into the beta cells.
The beta cell, this one is hard to tell because we did not do the linear tracing. We just traced a beta cell. We do find beta cell regeneration increase in the CF1. But for the acinar cell, whether this different duct cell, they maybe have. We have a hypothesis not to get the result yet. Is, for example, in the CF damage, the acinar cell will transfer from the metabolizer into ductal cell. This cell, I think they have the ability, maybe at some environment, they can stimulate from this cell transfer to the beta cell. Now we have a KRT7 drive the linear tracing cell. We want to see whether in the late stage or they have some beta cell come from the, not exactly the duct cell. I think it's the ADM cells from the church, but still not conclusion yet. So we have this plan. Hopefully next year answer your question. Okay, thank you.
I'd like to thank all of our speakers for such a wonderful, and all of the audience for a great discussion. Thanks so much.